In this experiment, we used fruit flies to violate the assumption of Hardy-Weinberg and their population size. By violating Hardy-Weinberg, we are basically bringing a drastic change in genetic diversity because of a smaller population. We created two breeding populations, one with one wild-type female and one white-eye female, and the second with five wild-type females and five white-eye females. We then keep the vials at home at a room temperature so the flies can reproduce, and every week we bring them to class to record phenotypic ratios.
In terms of the background of Drosophila fruit flies, the flies we are using are wild-type and white-eyed flies. The flies are good for fast reproduction that allows us to observe limited genetic diversity because of the small population. We also use primarily pregnant females for the first generation to allow us to get guaranteed larvae for the next generation. Throughout this project, we might see an ongoing competition between white-eye and wild-type flies, primarily due to the fact that these two phenotypes are going to be the main phenotypes we see during the project. There could also be possibilities of incomplete dominance or codominance. By limiting our population size to the point of violating Hardy-Weinberg's equilibrium, we will possibly see results from the bottleneck effect where we see a drastic change in the population size because of outside influences. From the bottleneck effect, our group believes that we will see different results between the 5 to 5 and 1 to 1 ratio because of the amount of flies in each tube, and that also the wild-type flies will also be the most frequent during the project. Throughout the project, we should also see an increase in the amount of flies every single week we examine the flies. This is also the citation for the photo that we used previously.